Okay, thank you for the introduction. Hopefully my slides will show up. This has been a great meeting for me so far. I've learned a lot. I really enjoy I'm very thankful for being invited, and I've enjoyed all the talks this morning. Got some notes in my notebook that I'm going to bring back to Michigan and try some of the ideas you guys have stimulated in me. So I'm going to tell you a little bit about retina regeneration using zebrafish as a model. As you probably know, let's see. Did I do that? Sorry, guys, this is, I was warned, it's a little sensitive. So as you know, the retina is a thin piece of tissue lining the back of the eye. It's composed of 6 neuronal cell types and 1 glial cell type that are laminated into 3 nuclear layers. So in the outer nuclear layer are the photoreceptors, the rods and cones. They sense the light. They pass the information to interneurons, amacrine cells, bipolar cells, and horizontal cells that then transfer the information to ganglion cells in the ganglion cell layer. And then through their axons, they form the optic nerve that connects to the brain. There's 1 major glial cell type in the retina called the Müller Glial. That's a radial glial cell type. It's the only cell type that sends processes radially to span all the layers of the retina. It also sends processes laterally and wraps neurons. And this sort of makes sense for a cell that needs to sort of monitor the environment and respond. And Müller Glial are involved in maintaining homeostasis in the retina. So when there's damage, they release factors that help protect and keep the neurons healthy. You can imagine what happens when neurons die in the retina. There's a number of neurodegenerative diseases, and they result in blindness. Oh, man. Sometimes I feel like I'm going forwards, and other times I'm going backwards. There we go. So they'll lead to blindness, and macular degeneration, glaucoma, and diabetic retinopathy are the leading causes of blindness in the developed countries. So macular degeneration is loss of photoreceptors. Glaucoma is the loss of ganglion cells. And in diabetic retinopathy, hyperglycemia results in a variety of neurons lost. And you can see that blindness starts initially in these cases with macular degeneration. The central retina, that's where the highest density of cones are. It spreads peripherally. Glaucoma starts in the periphery where axons start dying initially in the periphery, it spreads centrally. And then diabetic retinopathy is just random neurons dying throughout the retina. There's lots of approaches to treating blinding eye disease, and these include prosthetics. So you wear glasses that have a camera on it, and then through a wire, so it's the visual information, the light is transferred to electrical signal that's then transferred via a wire to electrodes that are planted behind the retina and stimulate remaining cells. That's something that people are using now to treat blindness. Sort of a biological analog of that would be optogenetics, where you would deliver a light-sensitive ion channel, like channel rhodopsin, to the remaining cells, and that allows them to sense light and transduce that information. If it was a single gene that was responsible, one could use gene therapy. AAV is a delivery vehicle. The diseases I just described, there's no genes that have been identified that actually cause those genes. They're probably either multigenic and environmentally resulting from. Stem cells recently have started being used as an approach towards treating blindness. And in this case, the idea is to take the fibroblasts from a patient, turn them into induced pluripotent stem cells with the Yamanaka factors is one way, and differentiate them in tissue culture, and then transplant those back into the eye. And the hope is that they'll integrate, wire up, and then restore sight to people that are missing those. And clinical trials have already been done with the RPE. There's a retinal pigment epithelium that lines the back of the eye. They've been transplanted back, and in terms of safety, it looks like they're safe. We'll have to see if they're able to be functional in future studies. All these approaches involve introducing something foreign into the eye: cells, electrical materials, genes, et cetera. And I think we'd all agree what we'd prefer is for our retina to heal itself. Sort of like when we scratch the skin, you know, and all of a sudden it goes away and we look good. So, you know, this would be a wonderful thing to do. As people have said, you know, and you certainly know, the mammalian central nervous system is not really good at regeneration. And in the retina, there has not been identified in the adult retina in the adult mammalian retina, stem cells haven't been identified. But hope comes from zebrafish, where regeneration is robust. So this is an example of a needle poke through the back of the fish's eye so that we can damage all the layers of the retina. You can see within a couple of weeks, the tissue looks pretty good. It's been regenerated. And if we use Wabane to destroy most of the retinal neurons in the retina, they do regenerate, they do wire up, and the fish regains sight. So we get functional recovery. It's quite amazing. And it's fairly fast. Because of this robust regenerative response, and because the fish retina shares structure and function with that of mammals, same cells, organized, same functional properties, we think, and also because so many genes, approximately 70% of the zebrafish genes are shared with humans, we think they'd be a great model system for trying to understand the mechanisms underlying regeneration, and then suggest that that information would suggest new strategies for treating humans. So that's what we want to do. We didn't know what the stem cell was in the zebrafish retina when we started out, but we assumed there was one. And I had done a bunch of reading and came to the conclusion, a little naively, that you have to realize this was done around a time when there was no transgenome data about the fish's genome or the mammalian genome. So we were sort of flying blind, but I got very lucky. And I had chose to take a fragment of the tubulin promoter, and it wasn't even called tuba1A at the time, but it was a neural-specific tubulin. I cloned it and put it in front of a reporter, ultimately GFP, and made transgenic fish with it. And that fish, if you look here, this is a retinal this is a whole mount of the retina. You can see there's very little GFP, but wherever we poked it with a needle, we saw robust GFP expression. And if we look at a cross-section, the cells had the morphology of the Müller Glial. They spanned the retina, and they had expressed glutamine synthetase, which is a marker of the Müller Glial. And most interestingly, they incorporated bromodeoxyuridine. And at 4 days post-injury is when the proliferation is maximal for these Müller Glial. And we lineage traced them over 3 months, and we found they made all the major neurons of the retina. What I'm showing here is the regeneration of a photoreceptor, a bipolar cell, and a ganglion cell. But it makes all the major neurons of the retina, which is amazing. This is an asymmetric division, so the Müller Glial regenerates itself. The Müller Glial remains. It produces a progenitor that then goes on to reprogram. So a lot of work has gone into studying this from a large number of labs, and I'm sort of summarizing the work of a lot of people here. But basically, these are the steps that are taking place in the Müller Glial, we think at least, that are important. And I'm sort of short-circuiting, or not telling you a lot about the last step, the differentiation, because nobody's really studying that initially. We need to, and people will start paying more attention. But everybody's been focusing really on these early stages because we want to understand if we can a way to make our Müller Glial act more fish-like. So let me just walk you through this a little bit. So the Müller Glial are sitting in a sea of neurons. There's some microglia, macrophage-like cells that are interspersed in the what we call the plexiform layers. The Müller Glial end feet abut up to vascular endothelial cells, and these are the blood vessel cells that are running along the surface of the retina, or the surface of the retina. And then after injury, what happens is these neurons start dying and they communicate to the Müller glia to start reprogramming. That changes gene expression. As immune cells then start infiltrating and congregating around the dying neurons, we find that those immune cells are also providing further reprogramming information to the Müller glia. So they go through a two-step reprogramming process before they can actually divide. Then they undergo an asymmetric division, produce a progenitor that transiently amplifies and ultimately differentiates in all the neurons. So in thinking about, so before I talk about that, I wanted to mention in the mouse, this doesn't happen, right? In mouse, what happens is after injury is what's called a gliotic response, and it's characterized by Müller glia swelling and the expression of intermediate filament proteins. Now, building on work that we had done in zebrafish, Tom Ray and Bo Chen's lab have taken some of the molecules that we found and forced their expression into the mouse, into Müller glia in the mice. And one example is ASCL1A, or ASCL1. In fish, it's called ASCL1A, but ASCL1, Tom Ray's lab shown, he's taken to ASCL1 and the histone deacetylase inhibitor TSA, and he's shown he can get Müller glia to make some bipolar cells. And Bo Chen's lab has been able to, I'm sorry. And Tom more recently showed that if he combines it with 801, ASCL1, he can make ganglion cells and amacrine cells. Bo Chen's lab has shown that if he takes beta-catenin, which is also required, both these ASCL1 and beta-catenin are required for regeneration in fish. He's shown that in combination with transcription factors that drive photoreceptor differentiation, rod photoreceptor differentiation, he can make photoreceptors. So this is all cool and very important, I think, because it's beginning to show that the mammalian retina, the Müller glia are a little plastic. They can respond to factors, and I think we're at the beginning. The problem is this is a transdifferentiation event, which you all have been talking about all day. And Müller glia are actually important for the retina, and we're concerned about depleting Müller glia and making new neurons. But we don't know how many you can lose to actually be a problem. So, we need to do the experiments and studies to be able to figure that out. The other problem is these neurons are immature when they're transdifferentiated. They don't seem to mature very well. And they also have, when you start looking at the gene expression profiles, they also have characteristics of the glial cell still. So they're sort of almost a little bit of a hybrid cell. Nonetheless, they wire up. Tom's shown really nicely. They wire up and they can transduce visual information. So, you have to sort of weigh the advantages and the disadvantages. And if you can get enough of these cells, right now that we can't transdifferentiate a lot, but if you could, you may be able to actually be able to get some visual restoration, and that would be very important. So I was trying to think about what to talk about today, and I went through the literature and said, I'm going to try to intersect something that I'm doing with what people do in the, in the hearing or develop, the cochlear development or regeneration field. I found all these papers that focused on Notch and Mick, and I thought, maybe I could tell you a little bit about what we know about those in the fish. So let's start with Notch. So you all know what Notch signaling is. It's a ligand-bearing cell that has a Notch ligand, a delta-like ligand or jagged, that interacts with a receptor-bearing cell, and that sets in motion a series of proteolytic events that releases the intracellular domain of Notch, so it can go to the nucleus, combined with the DNA binding protein RBPJ, and the cofactors mastermind-like and P300. So this activates gene expression. In the mouse retina, HES5 can be used as a marker for Notch signaling. And Tom Ray's lab shown that the progenitors during development express Notch, have high levels of Notch signaling. And as development proceeds, Müller glia are born postnatally. And you can see that in the postnatal retina, Notch signaling gets restricted to these Müller glial cells and then gets depleted. And when you look at the regenerative potential of the Müller glia, and this is done usually in explant cultures where you look to see if these Müller glia can respond to modifications that would cause them to make neurons, you find that this postnatal stage where Notch signaling is expressed is when they're most malleable and can be changed to and have some stem cell function. If we look in the fish, what we find is that this is a transgenic line of fish that has multiple RBPJ binding sites and from a minimal promoter driving M-cherry expression. It's called TP1 NLS M-cherry, and it's also got a GFAP-GFP transgene to mark the Müller Glial. So this is a notch reporter, the red, and you can see that when the Müller Glial are born, they're expressing notch. They have high levels of notch signaling. And what's different between the fish and the mouse is it's retained into adulthood. So we thought that's interesting because we see regeneration in adult fish, but we don't see it in mature mice. So we wanted to pursue that, and we first checked, is it even involved? Does it change during regeneration? And you can see that if after a needle poke injury, notch signaling is retained in the Müller Glial flanking the injury site, but right at the injury site, you can see the BRDU positive cells, they've lost their notch signaling. It's been suppressed. Furthermore, if we overexpress the notch intracellular domain, we force its expression with a heat shock promoter, we can show that we can inhibit regeneration. So notch is important for regeneration. If we ask what are the ligands that are responsible for, what are the ligands responsible for this, we were surprised to find that it's actually that we identified vascular endothelial cells expressing delta-like 4 that followed the notch signaling profile. When we did RNA-seq, if we did single-cell RNA-seq, what we'd find is the VEs, the vascular endothelial cells, regulated delta-like 4 in parallel with what we saw in terms of the notch reporter expression. And it turns out that what's going on here is that after injury, these immune cells are activating the expression of some ligands that are involved in VEGF signaling and also some cell cycle genes, which are very interesting, I think. But that activation working through these VEGF receptors, FLT and KDR on the vascular endothelial cells, reduce notch signaling, and then these cells are then proceed to start dividing. So that's one of the signaling cascades. I'll warn you, there's like 15 others that we studied that also participate. So but they don't work like this. We don't know how they work. This is the first one we really figured out at least this amount of details. Throughout the talk, I'm going to be listing the people who participated on the side here. So, wanted to understand, we thought this was interesting, and we wanted to understand, what if we lowered notch everywhere? Even the Müller Glial, that didn't respond to the injury, that were flanking the injury site. Would this also let them all of a sudden, respond to and enter a proliferative response? So this is just showing in the notch responsive, the injury responsive Müller Glial have high notch signaling. You injure the retina, it goes down. You get this small patch of proliferating Müller Glial. If we now lower the notch signaling level with this pharmacological reagent, DAPT, this is going to now lower notch signaling throughout the retina. What we found is that we get an expanded zone of proliferating Müller Glial. There's more Müller Glial being recruited to an injury response than before. The whole retina didn't start proliferating, but we got a wider zone of proliferating Müller Glial. That, to me, sounded interesting. A lot of people in my lab said, so that's great, let's move on. I'm like, why don't we figure this out? Because lowering a threshold could be key to figuring out how to make our Müller Glial respond. I mean, maybe they just have such a high threshold, we can't lower it. So anyways, we decided, so how do I think, what do I think is going on here is the following. We poke it with a needle, the eye. There's a gradient of factors, injury responsive factors that are released with the highest concentration where the Müller Glial are responding. At these peripheral regions where the concentration is low, normally they don't respond, but we feel that we've lowered Müller Glial's injury response, so now they can respond to these lower concentration of factors. How do they do that? We thought that would be important to figure out, so we hypothesized that notch regulates chromatin accessibility and the expression of certain regeneration-associated genes. And we wanted to test that, and what we did is we used ATAC-seq to look at chromatin accessibility and RNA-seq to look at Müller Glial gene expression. Our Müller Glial are fluorescently labeled so we can easily fact sort them and characterize them. In addition, what we did is we wanted to do this in the uninjured retina because those Müller glia that were being recruited were not, were experiencing lower than injury levels of these injury-derived factors. And we used two conditions to inhibit notch signaling, DAPT and then a genetic dominant negative mammal that we expressed with heat shock. And what we want to do is intersect this information with the injury response and find out if any of these injury responsive genes were changing their chromatin accessibility and gene expression. So this is the injury response. You can see that out of 22,000 genes that we interrogated, there were about 4,400 that were differentially expressed. With chromatin looking at ATAC-seq, about 29% of these injury responsive genes had chromatin accessibility changing, and most of it was going to open chromatin. Okay, so remember, notch signaling is an activator of gene expression, and we were suspecting that that notch inhibition might be turning on genes and opening chromatin. Notch inhibition would be doing that, and that thereby activating regeneration-associated genes. When we looked at the DAPT, and the dominant negative mammal injured treated retina, which was uninjured, what we found was a very small percentage of the regeneration-associated genes were being regulated, only about 2%. That wasn't a lot, and sort of it was a little depressing because I thought, oh boy, I expected a lot more to be regulated. When we looked at the ATAC-seq data, though, we got about 30%, which is very similar, actually, and a large percentage. I think most of these genes actually were the same ones that we see up here. So this was exciting, and it implied the chromatin accessibility may be a major factor that's contributing to this lowering of threshold. However, we didn't give up on this 2% because we predicted a repressor was actually being regulated by notch. Notch activates something, but if it's going to end up opening chromatin, most likely, when you inhibit it, most likely then it's going to act through a repressor. And when you get rid of notch, that repressor goes away, then you can maybe turn on genes and open chromatin. We looked at, I think there was like 85 genes or something in this 2%. And we found two repressors. There were only two repressors. So that made it really simple. They both were suppressed after injury. We forced the expression of both genes. I'm just going to tell you about one of them, both genes, but HEY1 and NID2B. And we found that they would block ASCL1A, which is that critical transcription factor necessary for regeneration. And they also blocked proliferation, suppressed it. Most importantly, though, was when we gene edited the HEY1 gene, when we inactivated it through a CRISPR-mediated mechanism, we got back our expanded zone of proliferating Müller Glial. So we suspect that HEY1 is mediating this fact. Now we have HEY1 mutant fish, and we need to now find out who the downstream targets are, so we can really understand what's the mechanism of this lowering of regeneration. But this is a key finding, I think. So now I want to turn to the MYC story. MYC, so zebrafish genome went under a duplication event a long time ago, and that's why many of the genes in zebrafish, we have A and B versions. So usually one of those versions is very similar to the mammalian ortholog of that gene, and then the other version often diverges and does something else. This is the MYC family in zebrafish. So in humans, in mammals, there's three MYCs. You're familiar with CMYC, we just call it MYC. There's MYC-like, MYC-L, and MYC-N, a neuromyc. In fish, there's MYC-A and MYC-B, which are orthologs of the CMYC. There's MYC-like A, MYC-like B, which are the orthologs of MYC-like. And then there's MYC-N, which is the ortholog of the human MYC-N. But we got this MYC-H gene that's not an ortholog of any of those MYCs, it's just a new MYC. That's sort of cool. And that's something that I've always been interested in is, does this gene, because people always say, why does the fish regenerate? We don't. Well, maybe that gene duplication event had some importance and produced genes that have some important novel functions that could be important for regeneration. It's still just an idea, though. This is the single cell RNA-seq from the Blackshaw lab that we use for identifying the cells that are expressing these things. Our Müller Glial RNA-seq data said these genes, the MYC-H was the most highly induced gene, whereas MYC-B was expressed at a high basal level. And this is a single RNA-seq showing you that indeed, this is the Müller Glial population over here. This is the activated or reprogrammed Müller Glial. These are the neurons of the retina and vascular endothelia cells, et cetera. But you can see the MYC-H is highly induced, whereas MYC-B has high basal expression. So the first thing we asked was, what percentage of the genome do these things regulate? Oh, I'm sorry. The first thing we asked, getting ahead of myself, was, are they necessary for regeneration? Of course. So we CRISPR'd them. We looked at proliferation of the Müller Glial, and we find that indeed they're necessary for regeneration. MYC-B has less of an effect than does MYC-H, but they both reduce the ability of Müller Glial to respond to injury. We've done double mutants now, and you get almost no proliferation at all. So they're sort of additive in their effects. We then, asked what proportion of the regeneration-associated transcriptome might these genes be controlling? MYC is notorious for controlling large amounts of genes, when it's expressed. So we did the usual RNA-seq, and oops, here's all the data at once, but it's pretty impressive. MYC-B is controlling 43% of the genome. So basically, we're taking our wild type and mutant fish, doing RNA-seq, and we're just comparing the genes, and what the injury responsive genes in the MYC- fish that get dysregulated is almost half of the transcriptome. MYC-H affects about 17%. When you look for the enrichment of biological processes, what you find is a lot of ribosome biogenesis, translation, DNA replication, and mitosis in the injury responsive Müller Glial. When we look at those that are just regulated by the MYC-B gene family, what seems to come to the top of the list is the mitotic type chromosome replication, and lower are the RNA ribosome assembly and the ribosomal RNAs. If you look at the MYC-H mutant fish, they seem to preferentially regulate. At the top of the list is the ribosome biogenesis, and translation is a little bit lower there. This ribosome biogenesis and translational control makes sense. A cell's dividing, it's got to make a lot of proteins. It's interesting, MYC controls this. It's known in other systems that MYC does this, so this wasn't a great surprise. But in our system, we didn't know that this was occurring, and we wanted to prove it. So gene expression is pretty good, but we wanted to see if we could visualize it. So we took advantage of this puromycin analog that would allow you to use clicket type chemistry. Copper, we could attach a fluor to it at the end of the experiment. So basically the idea is you use this puromycin analog, inject it into the eye of the fish, we injure it, and then we wait a certain amount of time, and then we use a clicket chemistry to visualize what proteins, how much protein synthesis is taking place at any particular time. And you can see this is now a NMDA damaged retina. So we've damaged the neurons throughout the retina with the neurotoxin NMDA. And you can see in the control, there's very little protein synthesis, a huge amount after we injure. And we confirmed it was Müller Glial, and we confirmed it works with the needle poke injury. This is the protein synthesis after needle poke injury, and you can see it lines up with the Müller Glial processes. None of the MYC stuff has been published yet. There's all sort of pseudo preliminary data. We hope to get it done over the next year, finish this stuff over the next year. Importantly, though, we looked at the MYC-H mutant, and it's certainly reduced in its the amount of translation taking place. So these MYC genes are really important for translation. So you remember I told you notch suppression expands the zone of Müller Glial. And largely that was just discovered by accident. Seemed to me, if MYC's regulating 50% of the genome, it too should do something like lower its threshold. And sure enough, when we force the expression of these things using heat shock promoters, you can see we get an expanded injury response. We haven't looked too deep into this yet. We just got these data, but what we did do is, right away we said, wow, this is cool. Why don't we combine the two and see if they synergize? And let's see if they do anything in the uninjured retina. So we did that. And these are the animals. We put them in DAPT. We heat shock them. We don't really use fire. And then we pulsed them with EDU 3 hours before sacrifice. And what we found is something quite remarkable. First of all, this is the retina treated with DMSO, not much happening here. This is a rod progenitor. It has nothing to do with regenerating the retina. It's a precursor to rods. This is Müller Glial dividing when you treat, we force the expression of mix. So there's a little bit of increase in proliferation. And we actually see that also with MIC-H. So MIC-B and MIC-H can increase proliferation a little bit. DAPT doesn't do much, get a little bit of an increase, but not much. This is what was amazing. We got a huge amount of proliferation when we combined DAPT with MIC-H and MIC-B. We did not expect this. And in addition, it's concentrated in the dorsal retina. I don't know what's going on here. I don't know how how this works, but I'm intrigued by it. I want to warn you though, that MIC has two functions that it's famous for. There are three, but 4 or 5, but two very important ones are cell division. Another one turns out to be apoptosis. MIC's fail-safe mechanism, I think, is to drive, if you get too much MIC, you may be necessary to drive apoptosis. We are just beginning to look at that. So I don't know if that's part of the underlying reason. So what I'm going to do is stop here and just summarize what I told you. I told you the immune cells and dying neurons contribute to Müller Glial reprogramming. Müller Glial derived VEGF engages vascular endothelial cells to stimulate notch signaling and Müller Glial and regulates Müller Glial's quiescence and injury response threshold. MIC-B and MIC-H stimulate Müller Glial reprogramming, Müller Glial proliferation, protein synthesis, and progenitor affects the whole process. MIC overexpression in combination with notch inhibition stimulates proliferation in the injured and uninjured retina. And we think, and the reason I'm telling you this is, just before I left, we saw more apoptosis, neuronal apoptosis in those MIC-treated retinas. So we think maybe this is going to be related to apoptosis. We don't really know that yet, and we need to do a lot more work to find out. So it's a correlation at this point. I'll stop there and take any questions.
So really nice talk. You mentioned that you thought that the genome duplication that occurred in zebrafish might have something to do with regeneration. And I'm just thinking about in terms of hair cells, we know that they regenerate really well in amphibians and reptiles, which have not had a duplication. So I'm wondering what's the status for retinal regeneration in frogs and lizards, say?
So in the frogs, it does occur. Müller Glial can be engaged. Doesn't seem to be as robust, and it seems to be species, Tropicalis versus Xenopus dependent. I think there is, in one of them, there's like they're tetraploidy or something. So there's some unusual things in those animals also. So yeah, so are these genes even expressed in those species? Yes. So that MIC-H gene, turns out it's in frogs and in reptiles, even without a gene duplication event, maybe. So no. Yeah, and chickens, postnatal chicks can exhibit a very small and feeble response to injury. They'll make some new neurons, but not all the retinal neurons, and it's very small amount.
Hi, Dan. Great talk. I just wonder whether this MIC overproduced system will increase retinoblastoma or other cancers. Is that something that we should be concerned about?
Cancer in the retina. inducing a tumor. So yeah, I think it's something we should be concerned about. I think that's the reason why, turning these things on is one thing, turning them off is another. I think, we don't want to, the retina is a great place to study. It's compartmentalized. I think if you want to do a study and you're worried that something might cause cancer, got two eyes at least. If you treat one, you may have the other one left. But, in all honesty, I think it's an important consideration. Nobody would want to introduce something that's going to cause widespread proliferation without control. And MYC is not a gene that I don't think anybody would want somebody to put MYC into their body. The idea is to understand the mechanisms and downstream targets so that we can do things that are more safe. This is just a beginning.
I have a question. So for the first part, when you show that you insure the retina and it regenerates in the zebrafish, so all the cell types are coming back, even rod and cone photoreceptors?
Yes, all of the cell types come back. Now, if you talk about, so just to give you a quick example, bipolars have like 30 flavors. Ganglion cells might have another 40 flavors, meaning there's variations, subtle variations. We have not gone in and asked, is every single subtype of those cells regenerated? But yeah, every major retinal neuron type is regenerated. Okay.
Great talk, super interesting system. And Pamela Raymond was actually pretty heavily engaged in talking to us at the beginning of regeneration. I remember very well, helping us think about our system. And I guess I'm thinking about these endothelial cells, which are kind of a surprising player, and wondering what's their situation in the mammalian system. And I can't even really imagine what the distribution of the endothelial cells are. Do they happen to line up with the Müller Glial cells?
Yeah. So maybe I forgot to mention it. So the blood retinal barrier is formed by the Müller Glial end feet abutting up to the vascular. So they're in intimate contact. There's flow of information back and forth. And I think that makes a lot of sense then that they would be involved in signaling because of that intimate relationship that they have. In the mammal, there's blood vessels also, but still the Müller Glial form and feed onto those vessels and for the blood retinal barrier, just as they do in fish.
Hi. I was wondering, is there intrinsic difference between dorsal and ventral retina that could account? I just remember developmentally, wasn't retinoic acid?
So you're right. Exactly. There is a few molecules. Retinoic acid is one of them that form gradients across the retina, and you get then the dorsal versus ventral and other aspects, nasal, temporal. And this has to do with, the ganglion cells need to project to the appropriate parts of the brain, and they have these characteristics that are associated with whether they're dorsal, ventral. So there is information there. We just don't know which part of that information is involved in what we observe. And we're looking into that, though. And we're slowly testing those molecules that are already known during development. Right.
So in terms of like single cell analysis, you don't see like two population of Müller?
No, we don't. But you wonder, the problem with single cell, it's not a problem, but you can make a lot of things happen in single cell. So you could divide populations up, and you get to a point where it's dangerous because you don't know if you're really looking at reality anymore. So you're right. There could be populations, and one needs to maybe tease out the specific cell types instead of having all the cells there. We should do single cell on the ganglion cells and ask, can we identify the dorsal population? That's possible.
So I have a question. So what happens to glial cell? What happens to photoreceptor cells like rod and a cone when you block a notch or when you overexpress MYC?
Right. So you don't regenerate them.
Well, at the gene expression level, do they change anything or?
Sorry, at the gene, what about?
At the gene expression level, do they change anything?
Okay, I say you're saying. We did not look, so I don't know right. So if we target the whole retina with our That's what you're getting at. Yeah. Is there a consequence of manipulating these other non-Müller glial cell types? Good question. We are using now heat shock promoters and Cree so that we use heat shock LOX-P and Cherry LOX-P factor that we want to express, GFAP Cree, to get it to the Müller glia. We're getting similar results with the genes we tested. So the data I'm showing you is relevant to the Müller glia. And you're absolutely right, though. We have to be careful. And in our early experiments, we did not. We just did everything. And DAPT is everywhere. So we didn't look at it. I'm sure it's affecting all the cells in the retina in some way. Yeah, that's a good point. Okay, thank you.
Okay, our next speaker is James Martin. He's from Baylor College of Medicine. And again, we are going